So we're going back to lab 1 here. And in lab 1, what we're doing is, if you remember, we took the brassica rhabda. These are the fast plants that have been artificially selected over time to grow fast. And now we use them for great examples in genetic research. And we planted this individual. So before we got to planting this individual, okay, the parental generation, or the P1, was made of true breeders. Okay, so these only had the recessive allele. This only had the dominant allele. This is the recessive allele for the color of the stem, the dominant allele for the color of the cotyledons, the baby leaves. This is the dominant allele only, which means 2 chromosomes, right, for the purple stem, and of course recessive for the green baby leaves called the cotyledons. These guys crossed. You really don't have to do a Punnett Square. There's only what? Because you have a what? In this generation, what is, let's see if you're paying attention, the frequency of the P allele here for this trait? Yes, Patricia?
1.
Nope.
0.
0.
That's not P, that's Q.
Oh.
I meant that one. All right, you're right, Patricia.
What?
Yeah, I messed up. You're right. So this is the Q, right? So the Q would be, I think I was right. So I'm right. I think I got this right. So if this is the recessive allele, the Q is 1, the P is 0.
Yeah.
Did I not say that?
No,
That's what the P value was.
Okay,
If
the
there's
P.
no Q, then
This is the recessive allele, correct?
Yeah, so it's Q.
So therefore, P is 0 and Q is 1. If the entire population, right here, has no what? Has no dominant alleles, the
That
P
is
is 0. This is 100% of all here. Okay? Now over, so I did get that right. All right? Now in this allele, or this other trait, because we're talking about 2 traits here, we know that here is dominant, so here the P is 1, the Q is 0. So just getting you there. Okay, so any case, you can only have a what from this chromosome, 100% chance of getting the recessive allele, and that's why it has it. There's a 100% chance of getting that dominant, and then when you take what? Remember, think of meiosis. You're going to get one or the other, but because they're true breeders, you can only get that. That's why we use them. So in any case, you get a dihybrid. So it's important to realize that on the F1, all right, we had little a and L, okay, a and L. And then we had YGR, and then YGR small. What's this tell us? Well, this tells us that we have purple. Why? Because we have, it's a heterozygote, right? And then we have green leaf. Let's not forget what this means. All right? This is dominant for green. If we don't have green, it's yellow. If we don't have the purple, it'll be colorless. So we had a purple stem, green-leafed cotyledon. That's what we had in the first generation. Okay, now we're going to cross these guys. Okay, now let's go see in a Punnett Square exactly what we should expect. Okay, and this is what we're going to do now. So we're going to do a dihybrid cross. All right. Now, if I'm doing a dihybrid cross, let's think about this for a second. All right. What are my combinations? All right. For instance, if this, I'm taking a cross of this individual with itself. We're crossing this with another A and L. And that's what we did. We crossed our F1 generation with ourselves. Okay? So, what combinations can you get?
capital A.
Yeah, so let's make that, let's go make that. And some people have some trouble with this. So I'm going to take this off for a second. Okay? And we're going to do that. So let's start up here. Okay? So from the cross of this with itself, we should think about what combinations we can have. first of all, right, if this is the individual, if it's gonna make gametes, this represents the diploid, we've gotta cut this in half, okay? So, when you think about what you're gonna put in your Punnett square, okay, what are the possibilities? Okay, well, I can either get big recessive or dominant, okay? I'll start with just the dominant. So A and L. Okay? And now I'm going to go to this one. And I'm going to go dominant, YGR. Okay, that's one combination. All right, I'll stay dominant. Couldn't I also get little YGR? So if I have this one, I could either have the big YGR that went with it, or I can get the other. Let's go recessive, A and L. And let's do YGR, big. And then what else can I do? A and L, and then the other recessive.
So 8.
And then we do the same thing on the side, right?
Exactly. So those are all the combinations, possible combinations of gametes, right? That they have to have. Now let's go the other way. Okay? All right, so that same idea. I'm gonna put them, it's the same individual, so we'll do it A and L. YGR. Then we got what? A and L. Little YGR. Then we have A and L. Big YGR. And then A and L. There's our recessive YGR. Okay? All right. So let's do the cross. Well, you put these together, what are you going to get? You're going to get a homozygous recessive, A and L, okay? And then I'll do it like this. A and L, and then YGR. I'm going to have to write smaller. I will work this, I'll get this out. Just going to write smaller. A N L Can
we put it under it?
it's got to be it's the total thing. It's got to be this whole thing. YGR. It's a dihybrid. We're looking at two alleles. And then YGR, I think anyway. Okay. I still got to write a little smaller. Okay. Now, that's one genotype. Let's go to the other one. All right, A and L. A and L. Big YGR. Little YGR. Okay, next one is B and A and L. A and L. Okay, and then we're going to have what? Little A and L. And then we have YGR. And then we have what? Then we have A and L. Big A and L. Oh. Little A and L. Big YGR.
Put that
Little YGR.
Are you giving us homework over Thanksgiving?
Am I what?
Are you giving us homework over Thanksgiving?
Probably not.
That's a family
But we'll see.
family bonding.
Okay, next.
I think someone's trying trying to call you.
It's okay.
We're more important, right?
Absolutely.
Thank you. That made me feel good.
A and L, A and
I
L,
think that's you.
YGR.
It is. No, it's not. It's his phone. I did hear it.
Wait, why are we going so fast?
I don't know, but
YGR.
I think you're just going really slow. This because I'm doing it all.
YGR.
I don't think I'm doing this right.
Did I do it wrong? No. I think I'm good.
We're doing it wrong.
Great
Okay.
skill, right?
Okay. A and L.
I'm really slow.
Big A and L.
Thanks for that.
Big A and L. Okay, then I go YGR. YGR. Recessive. Big A and L, little A and L.
Mr. Brodsky,
Yeah.
you said YGR, and then I actually wrote L instead of R. Or-
Sorry, I feel bad now, I'm over.
That laugh is evil.
You're right, it was. A and L. Okay, YGR. And then big. And last one, A and L.
Oh my god.
Oh, my Buddha.
You want the last one on it?
My wrist is starting.
What
to
is
hurt.
going on?
A and L.
Hold on.
My soul.
A and L.
I don't have a soul.
YGR.
Honey, you're not one to talk.
Did you get the joke, guys?
Yeah, I'm
YGR.
laughing so
You're
hard.
ginger. Yeah.
Oh.
Personality trait. That ain't your soul.
A and L.
Shut up.
You'll
A
be
How
saying
can
and
personality
L.
just traits of rocks?
Huh?
YGR. YGR. A and L.
You're so fast.
Okay, big
Did you get this on
A and L.
He's making us
Okay, and
Wait, I'm like really far behind.
YGR. Oh, you should be able to look at your own, right? AGR.
I'm a robot.
Wait, stop talking to yourself because now I can't focus.
ABC. You should never tell me that.
1, 2, 3.
I'm done.
You're done.
You're Holly.
Yes, I'm not.
I'm tall.
I'm Connor.
We know.
Oh, no.
With an E, guys.
I just think of Theo. It just makes me
I
laugh.
laugh very loud.
I laugh at my own jokes and my family makes fun of me for it. And you know, like when that happens,
My friend does that. We all make fun of him for it.
like I find the joke so funny.
No one else does that.
I know.
I know.
It's sad.
YGR.
Connor, most people just find your jokes offensive.
Your family's reaction.
Well, those are the best kind of jokes. What happened, Charlie?
I'm just mean.
Oh, that hurt. That hurt.
Precisely.
That hurt.
You're only hurt because she's speaking the truth.
That's
Yeah.
true. That's so true. That's like motivational.
What? Is this
You're
crazy?
mixing them up.
I am not.
This is sad.
Ooh, I'm putting it up to you, Mr. Brodsky.
Oh my god. So, can you draw lines? It's like so hard to distinguish them.
Just do it yourself.
Yeah, exactly.
No, it's like Grace. I almost. You should work. You should really finish that.
What?
You should really finish that.
Why? Mr. Grotsky,
Yeah.
why does everyone have one of these fun papers and I don't?
What's that?
Why does everyone have one of these fun papers and I don't?
Because Holly came in later when I did it.
Yeah,
Now
Holly, you came in
I'm in the
late.
middle of something else. Here you go.
Thank you.
All right.
I'm
Mr.
done
Brodsky,
now.
I emailed you again about my grade.
Okay.
Oh, yeah. I didn't email him about that.
I am sorry. I'm trying to get all the
Check it out
individual
online.
things.
It's
Mr.
better.
Grotsky,
This
right.
year I'm working my tail off, so it's like,
You ever thought about getting a
It's okay, you
secretary?
should give us access.
Yeah.
Oh yeah, that's a good idea.
Yeah.
what?
Wait,
Give us a
Connor actually had a
password.
You
good
should give
idea.
us access.
What? About a
Okay.
secretary?
Hand it over.
Mr. Gossi, if I be your secretary,
No,
Can I
you
give
can't be my
myself a
secretary.
To give us access.
a
I mean, no.
monthly 100 on my test. Every month I get 100 on a test.
Oh my god, I finished before Mr. Grotsky.
What?
Yeah, 'cause
I
you
said
got
it
the
all.
race.
He wanted a award.
I went.
She wants a
What?
cookie.
You wanted a water or
What? She wants a participation trophy.
A participation trophy is just saying congratulations for losing, but
Useless.
don't.
All right, that was too much fun.
Huh?
Oh yeah. We really did not get to do that.
Yeah, we
Every
did.
Connor received a lot
We
of
did.
participation trophies.
No, actually, I didn't.
I have.
I never signed up for anything. My parents wouldn't let me do
I already
sports.
did.
But
Because I
sadly,
would get hurt.
it was mostly
Everyone's
for
feeling our
lab
now.
notebook.
second or third place, but.
Well,
Next week,
I have brain damage from sports.
I
so
probably
Maybe
have
Friday.
it
about,
now, so.
I got concussion on a swing.
I probably own
I
about
got concussion on a
70
swing.
medals
Like,
and
I was just a dumb kid.
Was that last year?
because I'm
Congratulations,
that.
Sarah.
Didn't you get a concussion last year? Or like the year before?
Remember when Gabby got a concussion?
Football.
Ryan like spun her by her
everywhere.
I didn't get one from football.
My brother broke his wrist because my other brother pushed him off the most round.
I broke my wrist because I tripped over my shoelace.
My brother broke his arm, and then my dad made him ride his bike.
Oh, my dad,
Brother
broke
also
his
crashed into a
arm.
golf.
All right, here we go.
on his bike.
Oh yeah, he came on like all.
So let's go find the purple phenotypes. Okay, so this would be purple,
So all the ones with capital A and L.
or you just have to have one, right?
Can we use a highlighter?
What are these color pencils?
Does anyone got a highlighter?
How many colors do we need?
Oh, we have art class coming in handy.
Oh
Dude, that's so true.
Good thing I kept my pencils.
Our class always comes in handy.
I know,
I
always
had to
do.
cut something out in Spanish today. Whipped out
I
the
don't have
scissors.
any space for this
I
stuff
didn't
that
get
she gives
scissors.
me. Like,
That's so messed up.
Well, Connor, I don't think you can be
that.
trusted with scissors.
I can.
Oh, are you kidding me?
Oh, the ink
Gotta
is
be.
too dark.
wait, where did we get those from?
Talents, which one's blue?
I'm the designer.
Okay. So we've got purple, okay?
Why did you?
Now we're gonna do green. All I needed was one what? Dominant allele to get purple. So now, put my purple over there because I can't tell between the blue. Okay, here comes my green.
Oh no.
Okay, here comes green.
You don't know when you're
I don't really know what's going on.
Already. Good thing I didn't highlight it.
Good thing, don't worry.
Oh, I can even see
We'll
them.
see anything wrong.
Capital YGR is green.
Yep, green cotyledon.
Use that.
It's
That's
like
the
little
baby leaf.
leaflets.
That
Don't
was
people
on your
do
last
that?
test that you obviously got wrong.
I was on our last test.
Yep.
I don't remember
What?
that.
I don't remember that.
You have
was
to
our
identify
last
parts of a flower or like a plant. and
Oh.
Oh, I do
Oh
remember
yeah, that's the
doing
part that I
that.
had no idea what it was, but then.
I don't remember.
I wanna do that one. Yeah, I just have a bad memory.
I know that one, yeah.
Okay,
I don't
so
remember
it's
horrific
crossing.
experience.
Anything with a capital YGR
I
is
just
green.
did.
Yes, my brain chose not to remember the test.
I used to have color
I
pens and I
can't
took
do
them out of
this.
here.
Fun.
Excuse me.
I
I'm
just
taking it.
did two of them wrong.
Okay, so,
Mr. Gotsky, I just did this whole thing wrong.
all right, so we can do it again.
Aw.
Because you're gonna put this in your lab. I expect this somehow nice and pretty in your lab, and you can color code it. But
That's pretty.
how many purple greens do we have? 1, 2, 3, 4, 5, 6, 7,
Here we go.
8, 9.
You got a purple- Anyone got a purple pen?
So we've got 9
Pencil.
purple green.
You're just gonna borrow a purple pen.
Wait. Okay. Wait, can I have a pencil?
Thank you.
Wait, you missed the
That's
green on
probably
the bottom, but it
Well, I need purple green.
There's the green.
Purple green. Did I miss a green somewhere?
Yeah.
Oh, yep, you're right, I did. Thank you.
Wait, so which
Stop.
ones are green? The YGRs that are.
YGR, all you have to have is one dominant allele.
Okay.
Okay, so 9 purple green.
Oh,
All right, now,
Oy.
Fooey is right.
Dude, that hit me in the face.
that went like right through your hands. That was funny.
All right, so we've got,
12. Oh, I got 12 purple.
so we got 1, 2, 3, 4, 5, 6, 7, 8, and 9 purple greens. Okay, now,
What if there's only purple?
well, we'll talk about that. Now, I need yellow. I don't think I have a yellow marker here.
I had yellow color pencil.
Does this come out?
No, that's the highlighter.
That'll work. Okay, so yellow.
Oh yeah, what color yellow is it?
I win.
I don't do it. Oh my
Lello.
God.
Mr.
Lello.
Goss.
What?
Yeah,
There's
Lello.
a highlighter
Come
on
on.
the small.
Grace, can I have your Lello?
Oh, come on.
No,
You-
it'll come
you
off.
did it
You
for
just
a
have to
second, but
spray
it's-
stuff on it.
I'll get it all.
Yeah,
It'll come off.
it'll come off.
I'm a chemist, I know how to take things off.
Do you guys remember the smell shoppies, like the smelly shoppies? They like smell like
Yellow.
pineapple?
1, 2, 3,
Okay.
4, 5, 6, 7, 8,
Woo.
9, 10,
Yellow.
11. Why do I have 12
Yellow.
purple?
Yellow.
Yes, we're only
Well,
counting
you did it
purple
wrong.
greens.
Huh?
Purple and green.
Oh.
Dumbo.
Huh?
Whoa, that's rough.
Oh.
Okay,
That's
There's
really
a cat in
insane.
here.
so we got purple greens. I'm gonna write them in, why not?
No, I have to use my yellow.
Wait, so what is yellow?
Purple green.
Hey!
So we got
Take it.
purple green,
Okay,
Purple
so
green.
that's recessive, right?
Lello's my favorite color.
Same.
Yellow is
Purple
Wait, so
green.
what's recessive purple?
Are you kidding me?
Purple green.
Oh my god! I just did the wrong thing again.
Can I borrow Grace. a color pencil?
I
What's the
seriously
color
need to
yellow?
stop.
Oh my god. Yellow.
Purple green.
Small YGR.
Small white zero, small one.
Can I borrow a purple?
Wait, Mr. Grotsky, what are the recessive A and L? Oh,
Okay.
I got you.
Recessive means colorless.
I only have blue, I'm sorry.
You're not gonna have a purple, okay?
Okay.
Lello.
Purple,
Oh my god.
We're writing it all in, right?
yep. Purple green.
What?
This is purple yellow.
Not gonna look very nice.
Purple.
Where's my yellow?
Do I have to write it all in? I know it's space.
You can do whatever you like, but in your lab, you're gonna have to write it in.
Alright.
Purple yellow. Purple green. Purple green, okay, sorry.
What's the A?
Purple green, okay.
Stop talking out loud. I can't.
It's his classroom, his rules. Right?
Yeah.
Except I'm not a good child and I break the rules.
Okay, purple green.
Yeah. I've never
No
been
one thinks
suspended
you're
though.
funny, no one.
Wait, so
Yeah,
then
notice how no one laughed, Connor?
No one ever laughs at your jokes.
Actually, that is false. Someone laughed at my joke 5 minutes ago.
Someone. Who's someone?
Mr. Grotsky.
No, it was Theo's joke.
No, it was my joke. The one, yeah, it was my joke.
It's not funny.
Okay, now, A and L. That is colorless, non-purple. So we'll just say non-purple.
Can I just
I didn't
write
have any
NP?
You can color it white, but I can't see it, right?
Can I write NP?
Okay, non-purple, green,
You can very much see one.
Yes, you can.
So there's 3 yellows, right?
non-purple,
No, you
Yes,
can't
I
see
do.
it. Okay, I'm just gonna do it black.
Can I just write like
Green.
non-P?
Yes,
Because I don't have
you
space.
can.
You don't know.
purple,
I'm writing
green,
non-P green.
purple,
And I'll
yellow.
write it on mine.
Non-P. Look at how crowded this looks.
There's 4 non-purple,
You can do
right?
it, mine.
But this doesn't add up.
And this is purple, no, non-purple, non-purple,
Yellow.
green,
And pink.
and then non-purple. I like that.
Yellow. There should be 3, right?
Yellow.
Green.
I got 4 purple,
Oh,
non-purple.
this is like really.
Mr. Crosby, what's your star sign?
By what
I am a
time?
Gemini, guys.
Theo, what are
I am a
you?
Pisces.
Huh?
Purple,
What's your sign?
Zodiac.
that's my sign.
Yeah, no,
No,
like
I
what's
mean,
your
like the-
zodiac
What
sign?
month are you born?
No, it's not gonna work. It's not gonna work.
It said his birthday was
Okay.
last
Alright,
week.
yep, okay. So
Oh, wait,
we
was
should
it actually?
figure out something. We have 11, we had 9,
I'm sure the
Purple
Nintendo will
greens.
have something about him.
got 9 purple greens?
There's nothing.
He's kind of YouTube famous.
Oh, that's
Not really.
I bet I can find an old video.
How many
that
subscribers does he have?
will
It's like
resurface.
7,000
So
something.
9 purple greens,
Also, what was the like?
Alright, so we've got
YouTube channel.
purple greens, purple greens, purple greens, purple greens, purple yellow. How many do we have there?
Guys, have to
Purple
write-
yellow,
I'm
we have
guessing.
Purple
Three.
yellow,
No,
purple
there's two.
yellow.
Wait, I wonder if other teachers
Purple.
like use Mr.
No, there's
Grotsky
3.
Purple yellow.
In class.
So I got 3 purple yellow.
Oh, yeah. Come on. I meant 2 of the other
What did
things.
we tell you?
So it doesn't add up.
Okay, so 9 purple green.
3 purple yellow.
2 purple yellow.
1, 2, 3.
Okay.
3 non-purple green.
How about a purple green? We got purple green. Non-purple green. Non-purple green.
Wait, it doesn't add
No,
up.
2.
There's
No,
one.
1,
Are you guys blind?
1,
What? Are you
2,
blind?
3. Bottom
3.
left.
2. There's
Oh,
3.
yeah, yeah,
Where's
non-purple
the-
green.
It's right
It adds
there.
up.
There is 3, yeah.
It's
3.
1.
Non-purple
That is
Wait,
yellow. This
what?
is yellow.
That's not
green.
yellow.
That's not yellow.
That's green.
Good job, Sarah.
No.
And the last one,
There's
1.
we got 1
Not purple.
non-purple
Can I borrow the green again? Is that-
yellow.
I used the other one.
That's a double homozygous what?
Recessive.
I have green here, so I have the wrong color.
Only one is non-purple yellow.
Right, that is the rare guy, right, or gal?
That's the
One.
ratio.
Double
the
recessive.
9
Oh my gosh, the ratio is what in a dihybrid cross? 9 to 3
It works.
to 3 to 1 ratio. If this is Mendelian, if
What?
we have what? Equal chance. And there's no what? Evolution. There's no driving force to select one or the other. There's no huge population. There's no mutations. There's what? No gene flow. Okay. It has to be in Hardy-Weinberg equilibrium for that to occur, now that we understand that. And of course, we have to also think beyond no evolution, mutation, Hardy-Weinberg equilibrium. Besides that, we have to also understand that this works only if the traits are able to separate what? Evenly. I'm over here, guys. There has to be the idea, are these traits on different chromosomes? The idea for Mendel was that these traits separated independently of themselves. He called that independent assortment.
Yeah.
Whether you get an A and L or YG is totally independent of each other. Okay, these things separate independently of each other. He didn't know about chromosomes, but now we know that there's only in humans. Now, in this plant, I believe there's 7 or 9 chromosomes. Okay, which means a lot of genes travel on the same chromosome, correct? Like humans, there's what? 46 chromosomes, or 23 homologous chromosomes, correct? There's 30,000 genes. So most genes travel, and we know that. How do we know that genes travel together? Well, there's some visible ones. People that are blonde tend to have what kind of eyes?
Blue.
Right, that trait travels in the same chromosome. People that have a lot of freckles tend to have what color hair?
Ginger.
Okay, what color hair? You know, so the point is, sorry, I didn't want to go there because
It's not even like true.
I know, but the point I'm trying to tell you is that those are called linked genes. So Mendelian genetics is that these things are traveling on separate chromosomes, independently of each other. And of course, we're in Hardy-Weinberg equilibrium. So we would expect a 9:3 to 3:1 ratio of our plants. So I do expect this, okay, somewhere in your lab, okay, when you write that. That's the ratio we expect. So now what we're going to do now is we're going to go see what our phenotypes are right now, okay, as I lost my, and we're going to see what kind of ratios. Now, of course, we're assuming what? We're in Hardy-Weinberg equilibrium, and we're assuming that there was independent assortment of alleles, or in this case, chromosomes. And I don't know where my top went. All right.
Wait, what is this?
In any case, I'll just
It's
sacrifice
also
the
the
red.
broken plexiglass thing that you broke.
Okay, so party people. Now hopefully you can see something there, and you had a dihybrid cross on your last test. So that was effortless, sure. Okay, so what we're going to do now is we're going to take a little friends. Now we did not have great germination in the second generation by far. We have two, so we'll take what we got. So we're going to have to, we're going to do this at, you guys at home are going to help out as well in trying to decipher what the 4 phenotypes are. Remember, we have 1 of 4. Okay, we know which one is going to be the most rare. The non-purple yellow should be the most rare for obvious reasons. Okay, then we have non-purple green, then we have purple yellow, and then purple green. That should be by far the greatest amount. And of course, purple green is part of the parental phenotype. Yes. So now what we're going to do is look at the F2 generation that we planted from our seeds from the Dihybrid Cross that we did in class, and tally up the 4 different phenotypes that have resulted, and eventually be able to run a Hardy-Weinberg calculation to see if evolution occurred between the 2 generations. Remember, we planted dihybrids, and now we're looking at the offspring from our Dihybrid Cross, and just like the Punnett Square that we just did, we're going to get 4 different phenotypes, and we're going to try to count from our plants that resulted from the seeds of the cross and get our number of individuals who fit the 4 categories. And from there, we will run a chi-squared for lab 1 to see if our ratios are 9:3 to 3:1. And also we're going to do in class today is take these ratios, or at least the individuals, and try to run a Hardy-Weinberg calculation to see if evolution occurred. So that's what we're doing. And I didn't show all the video here, and it wasn't necessary. Okay, so we're going to continue on. So purple, green, how many did we get?
23.
23. Okay, we're writing this down. Purple, yellow.
Within the ratio.
9.
Green.
I'm going to talk out loud.
9. How many non-purple green?
8.
Hey, yeah.
23.
And then how many non-purple,
Wait, that's a big number.
non-purple yellow?
4.
What is that second one say?
Purple yellow.
It looks like a stick with a
Okay.
9.
9.
The little loop
Okay.
on top.
So in lab 1, you're going to do a chi-squared with that total number to see if in fact that actually what is an acceptable 9:3 to 3:1 ratio. Okay, now.
I forgot how to do that.
Okay, well, you'll figure that out, but that's not what I'm after today. I want to do a little bit of analysis with alleles. Okay, so let's do this. Okay, and I take my calculator, and we're going to do a little bit of Hardy-Weinberg calculations. I can't start here, can I? But I can start here. Okay, what do I know about non-purple yellow?
Recessive.
Recessive.
Right, it is going to be A and L,
A&L.
A and L, and it's going to be what?
Little YGR.
Little YGR. Now, in order to do Hardy-Weinberg with P and Qs, I've got to pick an allele. I have 2 here. Which one do you want to pick? Do you want to pick the
YGR.
purple or the color? I feel like the color of the leaf is something we definitely know. I feel like there was a little bit of interpretation with the purple, correct?
YGR.
So let's go YGR, okay? So we're gonna go, big P is of course green, and that of course is YGR. And we're gonna go Q, of course, that is a yellow, and that's little YGR. Okay, now, in the F1 population, okay, what do you think the P's and Q's were?
Wouldn't it be 1? If you're comparing to what you said
Q
before.
is 1 and P is 0.
Well, they add up to 1, but when we had the F1 and we planted them, they were all purple green leaves. They were all dihybrids. Can you guess what the frequency of the individuals in the first generation that we planted?
100 dihybrid. 100% of dihybrid.
They were all what? They were all this.
50-50.
Right. Okay, and it was what? They were all this. So don't we have equal amounts of, since we're dealing with just that allele, don't we have equal amounts?
0.5.
So 0.5, 0.5. Shouldn't we have the same frequency in the F2 if we are in Hardy-Weinberg?
Yes.
Yes, so let's go see if we are. Okay? That's what I love about this class. We can keep looking at real data and connect it to things that we're learning. Okay, F2. This is what we're going to calculate. Okay, now, can't look at the rest, okay? But what I'm going to do here is go total alleles. Okay, now I can attack this a couple different ways, but I know that my jumping off point is always going to be my recessive. Okay, so if I saw yellow, okay, I'm not looking at the purple. Okay, if I saw yellow, okay, I know that that was a double. I'm going to find those individuals that are Q squared, correct? So let's go find my Q squared. My Q squared are the individuals that are homozygous recessive. So, to find that percentage, okay, or that frequency, I have to do a part over the total. Okay, so let's go find all those individuals that exist. Okay, well, we have 9, okay, and then we have what?
8.
Okay, and 4. So we have 13 individuals.
But didn't you say part?
Yes,
Oh, well,
because
no,
how many individuals do we have total?
No, I was looking at purple and.
What's the total number of individuals? Remember you said, hey, remember that worksheets we had? To have PKU, it happened 1 out of 10,000 times. This is happening 13 out of how many times?
44.
44, yeah.
Okay, 44.
They
So
said
what they just did is they just added all the individuals.
Wait, where did you get 13? Sorry.
I counted
9 plus
the
4.
yellow individuals here, and there was 4 individuals, we're counting plants as individuals,
Okay.
part of the population. So 9 plus 4 is 13 individuals that wound up being yellow. Okay? Which means they have to be homozygous recessive to carry that phenotype. That's the jumping off point.
Now, is this considered a small population?
Yeah, so we're going to see if we have some issues here. So who's got a calculator handy? 9?
0.295.
What was that, thank you?
0.295.
0.295 is equal to my Q. Okay, here we go. So party people, what's the square root of Q and the square root of 0.295? Well, it's Q equals what? Square root of both sides. Who's got that for me?
0.543.
What was that?
0.543.
0.543. So
Good work.
Good job.
the Q for the F2 population, okay, is 0.54, and of course the P in the second is what? 0.46, right? Did I do that right? Yeah. Okay, so we have from the F1 to the F2, we have, my friends, a change in the frequency. Okay, this means that evolution has occurred. What 3 things provide a mechanism for evolution?
Gene flow.
Gene flow, stop there. Did we have possibly bees coming in the window already carrying fast plant pollen from other fast plants?
Yes.
Chances are no. That is how we would have gene flow. We'd have to have bees coming in, besides the bees that we didn't kill, but use dead bees. So we'd have to have bees carrying fast plant pollen from other classes to do that. So we can get rid of gene flow. Yes?
Genetic drift?
Genetic drift?
Yes.
How about natural selection? Could there be natural selection here?
No.
Very little chance.
There's a
LEDs.
very
Because-
little chance
no,
we
because
place the
it's one
under-
generation.
Yeah.
One generation,
It happened over
okay, it's gonna have to go over, so the chances of natural selection being at play here is very, very small, minute.
You aggressively sprayed them.
Yes.
Wait, but it didn't even change on me.
It didn't, but it did change. So we could say, party people,
So small.
that genetic drift is probably the culprit here because we're dealing with 44 plants, or we're dealing with a small population. And what does that mean?
It could shift.
It could shift very easily. Flipping a coin 5 times, you could get 5 heads in a row.
Like that thing you showed us where it like.
Exactly. If you flip it 5,000 times, it's going to work itself out to being, okay, 2,500 heads and tails. But if you have a small population, okay, you could have a shift. So listen, 0.46, 0.54, what does this mean? This is really important. In the F1 population, there was an equal chance of every gamete being made being either recessive or dominant. That is now being pulled in one direction. Now there is a little bit greater chance of the gametes being made being the recessive. Okay? Little bit, and little less chance of being. And what could happen? In the next, if we were to mate these guys, which would be kind of interesting, wouldn't it? You probably should do that. Okay, see wherever this is going to go. I would suggest that possibly the next generation, this number is bigger, just by the fact that there is a what? Greater chance of rolling a heads on this one. Okay, but you don't know for sure. But it can swing opposite. So what was the P squared here? So it was let's do the Q is 0.54. So I'm going to type in right now, this is the fitness. Starting allele is A1. We're going to make this the allele 0.54.
Are we gonna have to do
Okay,
this
now.
on our own?
Okay, just now we're going to run
You
this.
better not.
Wait, what is it?
Now the population size was what?
44.
Oh, no.
44.
I swear to God.
Okay, let's run it.
It's gonna die out, isn't it?
What happened?
It overtook.
Not even in first 100.
By having such a small population and what? Being 0.54 meant that the next generation is probably going to have what? More recessive alleles? There was runaway, but that was just one run. Let's do it again. Clear it again. Let's run it again. Last a little longer. Clear again.
Wait.
Whoa! That time it did what? We lost that allele.
Yo.
What this is doing is showing you how many generations, if you run the what? With the idea that if I have 0.54 alleles or frequency of the Q, what's going to happen with small populations based upon the roll of the dice? Sometimes we lost the dominant, sometimes we lost the what? This is exactly what genetic drift is. You can't do what? It's probably going to do that more often, right?
Yeah, because it's 0.54.
Exactly, but not necessarily. Not necessarily.